we're trying to understand the fundamentals and how they go awry in cancer cells. So then once we understand that, now we have something to target more specifically. My name's Alisa Piekny. I'm an associate professor in Department of Biology, and I'm also Tier 2 Concordia University Research Chair in Cancer Cell Biology. I would say that I have probably been incorporating synthetic biology in my research for my entire research career without realizing I was doing synthetic biology. I see it as taking information or material that may exist in one type of system or organism and then swapping it maybe into a different system or organism, or modifying an existing material within a system, creating new material within a system, and then using that in a way to understand fundamental functions. So understanding how that cell or system or organism actually works, right? Mm-hmm. So these are beneficial tools that we've been using for many, many years. Mm-hmm. We may have been doing it in a different way, where we've been doing a more classic genetics approach, but in that approach, we're modifying genomes. Mm-hmm. So when we use CRISPR, this fancy new genome editing tool, well, it's also editing genomes, mm-hmm. just like the way we were doing genetics before. Mm-hmm. It's just this way it's a little more targeted. Mm-hmm. So we can specifically mutate the DNA at a desired site rather than hoping to get it at the site that we're looking for. So, I mean, my research is obviously in the health sector, so I'm really using these tools to understand fundamental processes, say underlying how a cell divides, and then by understanding that information, we can generate drugs that target that process and that can be used to treat cancer, right? So I think in terms of the benefit of synthetic approaches, I see it from my eyes. I can understand the health benefit the best. I think if you, again, let's go back to the genome editing example. If you can imagine a patient that has a mutation, a very specific gene that's causing that particular disease, imagine if we could go in and edit that, right? We could actually cure somebody of a disease rather than treating them just for the symptom of that disease. Another way that I see the future of synthetic biology heading is, say you need to have a liver transplant. Well, right now that's very challenging to do. You need to match donors really well. Well, imagine if we could grow a liver in a lab that's pre-matched, right? So now you're not waiting for a patient to match and then to get that, to do that particular transplant. So I could see it being transformative in health. But, and this is where the public, I think, needs to understand that we realize that there's still a responsibility with how the research is being developed. And so there's policies in place to help ensure that we are responsible and how we're moving forward with this research, right? So that it's done in a very ethical way. So I would say that that should alleviate stress for the public to know that we are being socially responsible. I don't think we're that far. I mean, you know, this idea of genome editing in precise ways, or that, and understanding, I guess, how networks are controlling some of these pathways that would mediate things like, say, tissue rejection. I think combining those tools, right, is something that we never had the ability to do before. And so now I think we're at a place where that doesn't have to be that far in the future. But again, as soon as we talk about biology, it's very complex. Right. And so I can't predict how far in the future that would be, because for everything that we understand, there's about 1,000 things we don't understand. And so we could still be far away for reasons that we really don't know yet. But I would like to say that we're closer than we've ever been. It's that we're studying those things so that we can perhaps develop tools that could be used as therapies, or applications that could be used in a therapeutic way to help, say, treat cancers. And so cancers, yes, two of the, we call it hallmarks, are uncontrolled proliferation and ability of cells to migrate from their primary location to secondary locations. So we're trying to understand the fundamentals, again, the fundamental processes underlying those things. and how they go awry in cancer cells. So then once we understand that, now we have something to target more specifically. And so part of my program has actually gone there in terms of the application side by partnering with a medicinal chemist, and now we're designing compounds to specifically target components that are involved in helping to regulate cell division, for example. And so we can do this in a very targeted way, and by understanding the biology and marrying it with the medicinal chemistry, we can come up with drugs that are very highly targeted and hopefully will be more successful in the clinic, but let's see, that's a far way. In terms of the tools in general that we use in our research, we do things like genome editing, right? So we would use CRISPR, for example, to introduce a fluorescent probe into the genome so that we could visualize where something is locating within a cell. We can use it to generate mutations in specific locations so that we can understand, again, how that particular component works, and in a very specific way. We would use microscopy, and we use quite a bit. That's my primary expertise, is more on the microscopy side, but we would use things like confocal imaging, turf microscopy. These are all fancy terms that nobody would know, but what it means is that we can achieve high spatial resolution within a very, very small space, like a cell. So we can see very precisely where things are going, and more importantly, when they're not where they're supposed to be, right? So other tools that we might use are more biochemical techniques and in vitro, where we can look at how things interact together. And that's how we design the mutations that we make in the cell. So we look in the test tube and we see how things are working or not working properly. That guides us to where we should be mutating within the cell so that we can perturb those things more specifically. So yeah, that's it in a nutshell, I guess.
Awesome and complete answer.
Yes, so I mean, here is where we do biochemical work, we do molecular work, but of course we need to do a lot of work in cell cultures. We also have microscopy rooms, so these are dedicated rooms where we have the microscopes, again, they're quite sophisticated, and they require dark to be used properly. They require low vibration, so we have to have dedicated spaces for those too. And then we have the foundry, of course. And so this has become a very important tool for us as well. And so when we want to screen, for example, when we edit cells, and we want to screen large populations of those cells, or when we treat them with cancer drugs, and we need to screen large populations, yeah, we need to use the foundry for that work. That's very unique to Concordia, yes. And so the ability to do something like that combined with the other work, which I would say is maybe more traditional for many universities, I think that's what makes us stand out. Yeah. Okay.
That was awesome.